Okay, good afternoon, everyone. Thank you so much for the introduction. So as she mentioned, I will be giving a talk on the genomic diversity and antimicrobial susceptibility of invasive meningococcal and invasive Neisseria meningitidis in South Africa during this period of 2016 to 2021. Okay, this must be it. All right. Okay, so for those who are not familiar, Neisseria meningitidis is a human pathogen, so it's exclusive to humans, and it's transmitted through respiratory droplets. It is an opportunistic pathogen in that it can exist as part of the commensal bacteria in the human oropharyngeal cavity, which is what we call carriage. And it can progress rapidly to cause invasive disease, which includes meningitis, bacteremia, as well as survivors suffering long-term sequelae. And then the disease can also lead to death in a matter of hours. Okay, sorry, I see some differences in slides. I am so sorry. Okay, let's continue. Okay, so now coming to the invasive meningococcal disease in South Africa. So what we do know is that there is low incidence in carriage as well as disease, and that 17% of cases in IMD are fatal, as well as 20% of individuals suffering sequelae if they do survive their infection. And then there's high prevalence in the disease in individuals living with HIV, and then it's also mostly prevalent in infants, children below the age of 1. Treatment includes antibiotics, so we use cephalosporins in order to clear, or we administer cephalosporins in a case where meningo has not yet been confirmed. And then once the bacteria is confirmed, we use penicillin. And I'm going to be talking more on penicillin going further. And then rifampicin and fluoroquinolones are used to eradicate carriage in close contact. Vaccines are available. The quadrivalent vaccine that covers A, C, W, and Y serogroup. And then these vaccines are actually not administered routinely. It's reactive. So, and it's only recommended in high-risk populations, such as people living with HIV or people that are traveling to high-incidence regions. And then, as you will see going further, although serogroup B, men B, is one of the abundant serogroups in South Africa, we recently just licensed the vaccine, but it's not yet readily available to the general community. So now I'm just going to speak a bit about the incidence. So this graph here shows the incidence of meningococcal disease in the past 20 years. So as you can see, there's a general decline. It's my laser. Okay, I can't see it. A general decline. And so that's the dotted line, the black dotted line, in incidence, and this was also majorly caused by the decline in serogroup Y in orange, following the outbreak that was experienced in 2006, that high peak over there. And then another event that we noticed was the disappearance of serogroup A. Serogroup A, which was in purple over there, and the last detection of that was actually in 2010. And all this, mind you, is in the absence of routine mass vaccination. So for this talk, I'm going to be focusing on this period, 2016 to 2021, which is my research period. And then just to mention also that in the years prior to that, where antimicrobial resistance has been surveilled with the surveillance system, as my colleague has described. And we saw that on a yearly basis, the intermediate resistance to penicillin was just below 6%. So we were looking to investigate if that has changed in the recent years. So this here shows the global scale molecular epidemiology. Serogroups are actually endemic in certain regions globally. As you can see, I've mentioned, actually you will see in the coming slides, serogroup C, serogroup B, W, and now Y in South Africa, which is different from other areas. And then in addition to serogroup, which is based on the capsule, is based on the bacterial capsule. We have clonal complexes, which is the equivalent of a lineage. So clonal complexes are based on the differences in sequence types. Okay, and these are also distinct in different regions globally. So we have in the past described the molecular epidemiology of South Africa, and this was in this period, which is in the early 2000s. So we know actually that serogroup B, C, W, and Y are endemic in South Africa, and this is the correlating clonal complexes or lineages. And then in addition to serogroups and clonal complexes, we do also look at outer membrane proteins. And these are antigenic, which means that they are used are targeted for vaccine, production. So I'll be talking today on the PHA and the PHA proteins. There's a lot more, but for this talk, I'll be targeting that. And these are, I don't expect you to remember the numbers and the digits, but these are based on the prevalent clonal complexes within these dominant serogroups. So now, obviously, this was quite a while ago, and there is a knowledge gap. And in as much as there's not been any vaccine interventions, Neisseria meningitidis caused disease is it is a notifiable disease, notifiable condition in South Africa. And so it is important to surveil using technology such as whole genome sequencing. So now the aim of my study was to describe the molecular epidemiology and antimicrobial susceptibility of invasive meningococcal isolates, which we collect through the national surveillance system in South Africa in 2016 to 2021. And this is in line with the documentation that is from the whole genome from the World Health Organization, whereby they explain strategies towards defeating meningitis by 2030. And one of the overlapping pillars there is disease surveillance, in which I think this study fits in very well. So I'm not going to explain much on the national laboratory-based surveillance, as my colleague has already explained. You can remember this map over there. But the workflow is the same for IMD surveillance. And so from 2016 to 2021, we had a total of 585 cases. And from those, we received samples of about 82%, where we were able to use antiserum to detect serogroup and also confirm that using PCR. And then from those, we were able to grow cultures for 55%, in which we were able to do antimicrobial susceptibility tests. And then we were able to do a whole genome sequencing for 95% of those. And we used here the Illumina NextSeq platform. And then this is my data analysis workflow, where I used Trigonometric to for cleaning of my raw sequences, and then assembled using spades, so de novo assembly. And then I used 2 pipelines for my analysis. Papamillis-T Neisseria database is widely used for the majority, really, of Neisseria meningitidis studies, and it is able to curate and detect lineage, PHA, FET-A, PIN-A alleles, and even more. So it's quite a useful database and repository. And then I also use the BM-GAP pipeline, which is under the CDC, for mutation detection within the PIN-A. For my penicillin analysis. And then I also went ahead and to confirm this with an alignment using the PIN-A gene. All right. Okay, coming to my results. So this is a representation of the population structure of my isolates. So I just chose to show a basic tree where I color-coded the serogroups. So you will see that serogroup B accounts for close to half of our isolates there in blue. And then we found 23% of serogroup W, 18% of serogroup Y, and 10% of serogroup C. And then we can see there that there's very high diversity of serogroup B, where there's lineages of serogroup B, and then as opposed to serogroup W there, which only is characterized by 2 lineages. Okay, so I'm not gonna spend a lot of time here, but this is just to show the diversity of PHA variants among dominant serogroups, so B, C, W, and Y, and also by the dominant lineages or clonal complexes. So here we wanted to see how diverse these variants are as opposed to or as compared to what we've detected in, 20 years ago, about 20 years ago. And then we've noticed that, yes, there is a very high diversity compared to then, and also that the variants are not very different, but we did see or notice an antigenic shift in serogroup Y, where, and I'll explain this further in the summary, but the clonal complex 175 was the major cause of serogroup Y. It accounted for around 80% of our isolates, whereas in this study we see only 10% of this lineage. And now the leading cause is clonal complex 167. Same result with the FET-A variants. We see the same or similar variants and a higher diversity compared to previous years. Okay, now coming to the antimicrobial susceptibility testing. So this is the phenotypic results. And this just shows the distribution of what was intermediate resistant versus what was susceptible. And this, yes, I mentioned this is penicillin. And we can see where the intermediate resistance isolates are distributed. So serogroup B, W, and Y, as well as non-groupable genotypes. And then this is now looking into the allele, pen allele distribution of what was intermediately resistant to penicillin, which is a total of 40 isolates. And this makes up around 15%, more than 20% really, of isolates, as compared to what we've been seeing prior to 2016, which was a low 6%. So we can see here that there's not really a responsible lineage, but we do realize, notice that allele 9 and 8 are in most of the isolates. Okay, to wrap up, my time is up. Okay, so we saw from the study that serogroup B, W, Y, and C were the leading causes of IMD in South Africa in this period, 2016 to 2021, which was observed in previous years. Of course, with waxing and waning along the years, as you've seen in the line graph of incidence. And then lineages are concordant with previously described isolates, as I've just mentioned. And then the prevalence of clonal complex 175 in lineage in serogroup Y isolates decreased from around 80% in previous years to 10 in this current study. And now the leading cause is clonal complex 167, which is up to now 52%. And then further, this lineage, the leading lineage in Y167 isolates, is now characterized by sequence type 884, whereas in previous years this was 167. Right. And then intermediate resistance in penicillin continues to be driven by the PEN-A mosaic, which is where we detected 5 mutations in the PEN-A gene. The proportion of non-susceptible bacteria has increased from around 6% to more than 20%, as mentioned. And then common alleles were PEN-A and PEN-A9 and PEN-A14. So just to conclude, genomic surveillance is a powerful approach to monitoring changes in genetic profiles of invasive meningococcal strains, as well as understanding mechanism of non-susceptibility towards antibiotics used in South Africa. Okay, I would like to thank the following groups, as well as my supervisors, as well as PHA4GE for allowing me to speak today. Thank you.